MS Ramaya University of Applied Sciences.
Hi viewers, let us discuss on colorimetric estimation of copper. Before performing the experiments, let me tell you the applications of colorimetric estimations. Colorimetric estimations are useful in food industry where colorimetric data can be used to adjust the color components in a prepared food or beverage recipe to improve eye appeal, to gauge doneness in a baked product, and in fresh foods to determine factors such as degrees of ripeness and spoilage in relation to shipping, storage, shelf life, palatability and disposal cycles. Biological culture. Colorimeter can be used to monitor the growth of a bacterial or yeast culture. Laboratories. Used to test for water quality by screening for chemicals such as chloride, fluoride, cyanide, dissolved oxygen, iron, molybdenum, zinc and hydrazine. Used to determine the concentrations of plant nutrients such as phosphorus, nitrate and ammonia in the soil or percentage of hemoglobin in the blood. Colored solutions have the property of absorbing certain wavelength of light when a monochromatic light is passed through them. Amount of light absorbed or transmitted by a colored solution is in accordance with Beer-Lambert's law. According to Beer-Lambert's law, amount of light transmitted through a colored solution decreases exponentially with increase in concentration of the colored solution and increase in path length of curvature or thickness of the colored solution. A is equal to epsilon Ct, where A is absorbance, C is concentration, T is thickness and epsilon is a constant called molar extinction coefficient. It is constant for a given substance and wavelength of the color of the solution. The instrumentation involves a light source, a lens, a monochromator, sample holder, detector, amplifier and readout display. Procedure. First, let us transfer a copper sulfate solution, stock solution to a burette and draw out 5, 10, 15, 20 and 25 ml of the copper sulfate solution into 50 ml volumetric flasks. Now collect the test solution from the teacher examiner. Add 5 ml of ammonia solution to each of the above solutions and dilute up to the mark with ion exchange water. Shake well. Stopper the flasks and mix the solutions well. After this, prepare a blank solution. For this, take 5 ml of ammonia solution in a 50 ml volumetric flask and dilute it up to the mark with distilled water and mix well. After 10 minutes using blank solution, set the colorimeter to 0 absorbance at 620 nm. Measure the absorbance of the standard solutions against blank at 620 nm using a colorimeter. Now, measure the absorbance of test solution and tabulate the readings. Draw a calibration curve by plotting absorbance against concentration of copper. Using the calibration curve, we can find out the concentration of copper. Colorimetric estimations have limitations also, like similar colors from interfering substances can produce errors in results. Beer-Lambert's law deviates for dilute and highly concentrated solutions.
Hi students, today aim of the experiment is to find out the amount of copper in the given test solution. To find out that amount of copper, we are using the instrument called as digital colorimeter. This experiment is based on Beer-Lambert's law, which says that amount of light observed is directly proportional to the thickness of the media and the color intensity, or I can say, or the concentration of the solution. The apparatus required to find out the concentration of the copper sulfate, the given copper sulfate, is the digital colorimeter. The digital colorimeter consists of the set 0 button, where using the blank solution you have to set the instrument to 0, or if you are testing the transmittance you can set it to 100% by setting this set 100 by using this knob set 100, and this is to set towards the transmittance or towards the observance. This mode can be situated. First, now it is in the transmittance mode. If you press it once, it will be going towards the transmittance mode. So the next one is about this filter. So you have to set the filter to 620 lambda maximum, that is for the copper solution, the blue colored solution. We have 600 and the next filter is 720. So you have to set it to 600, which is nearby the value of the lambda maximum what we are going to use it for this experiment. a series of standards to prepare the series of standard solutions. So we require a series of standard solutions to find out the concentration of the given test solution. So series of standard solution using a standard flask. So we have a 100 ml standard flask. So this is the mark. You can see the mark. So up to here you have to make. So series of standard solutions means, so you have to take 5 ml, 10 ml, 15 ml, 20 ml, 25 ml, and 1 will be for the test solution. So 5 ml of the copper sulfate and 5 ml of ammonia you have to fill and make it up to the mark using the distilled water, making up to the mark using the distilled water. So the same thing should be done for all of them. So if you are taking 10 ml of copper sulfate, then 5 ml of ammonia and make it up to the mark. So all 15 ml, 20 ml of copper sulfate, but in all the cases ammonia will be only 5 ml. And one blank solution you have to prepare, and that blank solution should be containing only 5 ml of ammonia, and the remaining should be with the it should be make it up using the distilled water. So 1 burette. So in this experiment we are using a 50 ml burette. So it starts from 0. So then we are going to use according to the required standard solution. So 5, 10 ml if you want, you can add 10 ml. The lastly if you want 25 ml also, we can use 25 ml. So it is a 50 ml burette. The copper sulfate. So this is the standard copper sulfate which you are going to use to prepare the series of standard solution. So and ammonia. So this is ammonia which we are going to add 5 ml for all the flasks and also for the blank solution to increase the color intensities, and to the blank solution to nullify the absorption of ammonia. The remaining is the distilled water. So distilled water, what you are going to use it will be always filled in the wash bottles that you can use for the making of the solutions. So these are the apparatus that what we require. Before starting the experiment, all the glass, the standard flasks should be cleaned and then washed with the tap water, rinsed with the distilled water, and then taken for the experiment. So the burette also should be washed with the tap water, rinsed with the distilled water, and rinsed with the copper sulfate solution, and then we have to use it for the experiment. So the standard flask should be washed with the tap water. Thoroughly washed with the tap water and then rinsed with the distilled water. So this washing should be repeated for all the standard flasks that what you are going to use. The burette also should be washed with the tap water. With the top water on both the sides, so you have to release it. And then rinsed with the distilled water. And then it should be rinsed with the copper sulfate, the given standard copper sulfate, what they have given to you, it should be rinsed with the given copper sulfate. So now the apparatus is ready. In the same way, you have to use another burette for ammonia. So you can ammonia burette also should be washed with water, distilled water, and then rinsed with the ammonia solution. So you have to prepare the standard solutions. To prepare the standard solutions, we are using 3 burettes. So one burette we are using it for the preparation, means to add the copper sulfate. The second burette we are adding ammonia. So ammonia should be added 5 ml for all the solutions. The test solution also 5 ml, and for the blank solution also you have to use 5 ml. So lastly the distilled water. The distilled water is added when you are means you have to prepare it exactly 100 ml. So the last few drops you can add it using the last burette. So you have to carefully remove it. So this burette is already rinsed with copper sulfate. So you have to add the copper sulfate up to the 0 mark. So little bit of air gap is necessary to add it. So okay, filled. Now you have to adjust it to 0 mark. So adjust it to 0 mark. So this solution, since it is diluted, it is not necessary to pour it to the standard solutions. You can pour it to the sink. The lower meniscus should be touching the 0 mark. So now it has been set it to 0. The second burette should be filled with ammonia. So ammonia you can fill it up in the second burette. So this should be washed with water, rinsed with ammonia, and then you have to fill it up. So it is diluted ammonia. So you can make it up to the 0 mark. You can make it up to the 0 mark. Carefully clamp it. So and the last one should be filled with the distilled water. This is not necessary to make it to the mark or anything, because this we have to add it to the final when you have to make it up to the mark using the in the standard flask. So we have to prepare a standard solutions. As I said, a series of standard solutions should be prepared. So series of standard solutions. So we have made up a mark here. So 5 ml, 10 ml, 15 ml. So you have to prepare. So you can mark it as you are using 5 ml of copper sulfate in this. So I have made it the mark as 5. So now I have to take 5 ml of copper sulfate to this solution. So 5 is here. So add it. To start from 0, add it up to 5 ml. Carefully you can hold it in the hand. The left hand should be with the mark. The 5 ml is there. So done. So you can adjust the finals one. So make it to the eye mark. The first flask I have filled with 5 ml. The second flask with 10 ml you have to fill. You have to fill it to 10. So it was 5. So now you have to fill it up to 15. So now we have transferred 10 ml to this flask. So now it is 15. So 5 ml was for the first flask. So now it is 10 ml. So you are getting 15. So you are taken to the second flask. Similar way, you have to take it to the third flask, 15 ml. So you can continuously go on adding it, 15. So next 30, then 20 ml, and then 25 ml. Now to all the standard flasks, whatever you have taken copper sulfate to all the flasks except the test solution will be given by your teacher. To all the flasks you have to add 5 ml of ammonia. 0, 5. So we are going to add 5 ml of ammonia. Okay. So one thing we can observe, as soon as you add the ammonia, the intensity of the color has been increased. So why it is increasing? So this is because it is forming a cuprammonium complex. So you have to make it up to the mark using the distilled water. So, up to the mark, this is the mark, you have to use the distilled water. Up to the neck, you can use the wash bottle. So, it is up to the neck. Now, to the mark, it is very difficult to control using this wash bottle. So, I am using another burette which I have filled with the distilled water. So, we can go on adding it in a dropwise fashion, slowly up to the mark, the lower meniscus. If you are able to see, otherwise you can take to the upper meniscus. So, it is better, you can see I am able to see the lower meniscus here. So, I can take to the lower meniscus. Still it is not, the line is still above the lower meniscus. So, I can add few more drops, 1, 2, 3 drops. So, now you can see I have made exactly the lower meniscus touching the mark. So, to the next flask, to all the flasks you have to add 5 ml of ammonia. So, I am just showing you once again the second flask, the 10 ml. So, to this also you have to add 5 ml of ammonia. So, it is 5. So, drop it to 10 now. So, we will be adding 5 ml of ammonia. So, I added 5 ml of ammonia. So, you can see, then this also should be made up to the mark using the distilled water. Up to the neck you can use the wash bottle, then from the neck to the above to the mark you can use the distilled water. Using the burette you can make it up to the mark, because you can control. Okay, now it is there, yeah, 1 drop still less, add 2 more drops. Yeah, I have made up to the mark. Now we have to shake it well for the uniform concentration. So, after preparing all the series of standard solutions, so you have to thoroughly shake it. So, you should be getting the uniform concentration in the 100 ml standard flask. So, now I have shaked uniform concentration. We can see the color intensity of this and this is completely different, because this is containing 5 ml of copper sulphate, 10 ml. So, next will be 15, 20, 25 and 1 you have to prepare a blank solution. The blank solution will be containing only the ammonia. So, you have to add 5 ml of ammonia to it. So, 5 ml of ammonia, to the blank solution, only 5 ml of ammonia, and you have to make it up to the mark using the distilled water. So, 2 drops. So, blank solution is ready. So, that also should be shaked uniform. Now, so the test solution will be given you by teacher, as I said. So, to that also, so if they are given you, you do not know the unknown quantity of the copper sulphate in that, to that solution also you have to add ammonia, make it up to the mark using the double distilled water. So, now you have prepared a series of standard solution, 1 blank solution and 1 test solution. So, all the solutions should be ready before starting the experiment. So, you have to switch on the instrument. So, you have to see whether it is in transmittance mode or in the absorbance mode. So, you have to press this mode button. So, you can see it is going to observance mode. Since our experiment is designed to carry out, means we have to record the absorption. So, I am going towards the absorbance mode here. So, this is the cuvette which you will be using it to record the absorbance of the given solution. So, this cuvette should be filled 75% and then you have to place it inside the colorimeter. So, while placing it, you have to see that you are placing it in same direction throughout the experiment. So, we can just make one mark and then go on placing it in same direction throughout the experiment, as our experiment is based on the absorbance. If the thickness varies, the absorbance also will be varied. So, you have to keep it in the same direction throughout the experiment. So, before starting the experiment, as I said, so solutions are ready for me. So, now using the blank solution, so you have to rinse the cuvette, should be rinsed. So, you can take 25% and then swirl like this. So, thoroughly it is rinsed 2 to 3 times. You have to rinse it. The cuvette should be cleaned before using it. So, you can rinse it using the solution that what you are going to use. So, I am going to use first the blank solution to set the instrument to 0. So, you have to wipe it. See that there are no fingerprints. So, it should be cleared. Now, I have to set the instrument to 0. So, before setting it, see that whether it is in the absorbance mode or in the transmittance mode or in the observance mode. Now, it is in the absorbance mode. Set the instrument to 0 using this set 0 button. So, I am able to set the instrument to 0. So, setting the instrument to 0 means you are nullifying the absorption of ammonia and distilled water. So, there is no absorption because of this ammonia and the distilled water. So, you are using this set 0 using this blank solution. Now, rinse with the solutions that what you want to, means rinse with the standard solutions. So, rinsing always it is there. So, 25 you have to use 10-15%. Twice you have to wash with the same solution that what you are going to use. It is not necessary to wash with distilled water each time. So, rinse, fill it up to 75%, thoroughly wipe. There should be no water traces or fingerprints. So, keep it in the same direction as what you have kept. The direction should be same. And note down the absorption for 5 ml of copper sulphate what you have prepared. So, 5 ml means it is standard solution. So, they have given it, they have dissolved some quantity, it is a standard solution. So, the concentration of the standard solution given to you will be given. Based on that, you have to calculate what is the amount of copper present in this 5 ml also you can calculate. So, now would note down the absorbance. For 5 ml you can note it down as 0.07. So, the next we have to discard this solution, take the next standard solution, rinse it, so wipe it thoroughly, place it in same direction and note down what is the absorbance for the second standard solution. So, like this you have to repeat it for all the standard solutions and lastly for the test solutions, and find out by using the graph what is the amount of copper sulphate given, means it is 5 ml, 6 ml, 8 ml, whatever the amount they have given to you. Using the graph you have to find it out. So, finding out that amount of test solution and reporting it to the teachers will complete your experiment.
At MSR UAS, we spark your imagination unlike any other.